Inflammation can be defined as a complex reaction to injurious agents that consists of vascular responses, migration and activation of white blood cells, and systemic reactions. It is the body's immediate response to damage to its tissues and cells by pathogens, physical injury, or noxious stimuli such as chemicals. Inflammation is a protective attempt by an organism to remove injurious stimuli and to initiate the healing process. The inflammatory response is a defense mechanism that evolved in higher organisms to protect them from injury and infection. Inflammation is characterized by the 4 cardinal signs and the disturbance of function. The 4 cardinal signs of inflammation are redness, heat, swelling, and pain. Inflammation occurs via 3 main phases. Phase 1, inflammatory response. Phase 2, repair and regeneration. And Phase 3, remodeling and maturation. During these phases, the process of inflammation contains and isolates the injury, destroys invading microorganisms and inactive toxins, and prepares the tissue or organ for healing and repair. Additionally, there are 2 types of inflammation, acute inflammation and chronic inflammation. Acute inflammation is a rapid onset occurring in seconds or minutes with a short duration lasting minutes to days. On the other hand, chronic inflammation has a longer duration and is associated with the presence of lymphocytes and macrophages.
Good day, my name is Shania Smith, and I'll be discussing the relevance of inflammation to innate immunity. Inflammation caused by infection and tissue damage is a critical innate immune response mechanism. Inflammatory responses protect against a wide range of infections and facilitate long-term adaptive immunity against specific pathogens. The inflammatory response is important, and it acts as one of the defenses our body has against pathogens. It is initiated once the first barrier of defense, the skin, has been broken or compromised. When a pathogen has broken the skin, the immune system immediately kicks in to fight off the intrusion. Many chemokines and cytokines are released, and as a result, vasodilation occurs. This vasodilation of the blood vessels causes swelling and redness due to the now increased blood volume and blood flow. This enlargement of the blood vessels is important because it makes it easier for immune cells to move from the blood vessels into the damaged area. One of the main immune cells that are heavily activated during the inflammatory response is phagocytes, which are cells heavily abundant within innate immunity. It is important to have this vasodilation so the influx of these phagocytes can be greater. These phagocytes readily digest and remove the invading pathogens as well as any compromised skin cells. Additionally, this vasodilation allows for many other types of cells to flood the damaged area. It allows B and T cells to migrate to the area faster. This is a way that inflammation aids in adaptive immunity. It also allows erythrocytes to flood the area so that healing and repair can also happen quickly. Other cells like antibodies and complement proteins are also allowed into the infected area. So essentially, inflammation allows for a high concentration of immune cells into the infected and damaged area. This high concentration is important to reduce and fight the initial infection. It is extremely important to have inflammatory responses. The immune cells that engage in fighting the invading pathogens are mostly generalized and have no specific or memorized method of destroying them. Inflammation is an occurrence that occurs with any and every invading pathogen. It is also because of its quick onset, instinctiveness, non-specificity, and limited potency that inflammation is heavily associated with innate immunity.
The relationship between chronic inflammation and type 2 diabetes. Insulin is a hormone that is made by cells in the pancreas. Insulin controls the amount of glucose in the blood. The body becomes less sensitive to insulin, and type 2 diabetes starts to develop. This results in insulin resistance and leads to inflammation. The pancreas has cell clusters, beta islets of Langerhans. These cells excrete insulin when glucose levels are high. Glucose is transported to the islets via the bloodstream, and the beta cells release insulin as a response. High levels of glucose therefore stimulate more insulin release. However, sustained release of insulin also leads to the secretion of another hormone-like substance from the beta cells. However, these hormone-like substances affect the white blood cells. The white blood cells consume it, and with combination with glucose, free radicals are produced. Sentinel proteins in the white blood cell unwind, activate, then aggregate with 6 other proteins forming a ring-like structure. This structure then binds to 7 other adapter proteins. This is the inflammasome. The inflammasome activates a protein, which in turn activates another. This protein is crucial in the inflammation of the body. For example, interleukin 18 is a pro-inflammatory mediator activated by inflammasomes. It is produced and released by human adipose tissue. This pro-inflammatory cytokine increases the production of other pro-inflammatory cytokines and exacerbates obesity-induced systematic inflammation. Systematic inflammation is categorized as high concentrations of inflammatory mediators and immune cells in insulin-dependent tissue. Type 2 diabetes, which is correlated to obesity, exhibits inflammation in skeletal muscles and the liver. Furthermore, the inflammation spreads to other tissues. Upon release from the white blood cell, the inflammatory protein causes the beta cells to die and islets shrink. Blood glucose levels are no longer able to be controlled efficiently. High glucose levels then cause many damages to different tissues in the body, which is a vicious cycle. So, the more glucose, the more inflammation, and the more damage to the islets.
Chronic inflammation is characterized by the buildup of inflammatory cells in the absence of any damage or threat. Infection, autoimmune illnesses, and long-term exposure to errants are some of the causes of chronic inflammation. Type 2 diabetes is an autoimmune disease, which means that the immune system mistakenly destroys its own cells, mistaking them for antigens. Obesity is a risk factor for insulin resistance, and through understanding the process that links obesity to insulin resistance, it will better help us to understand the type 2 diabetes pathogenesis and also how to treat obesity-related illnesses. During obesity, A2 tissues, are also known as F cells, remodel triggers a slew of internal and extract signals that might activate an inflammatory response. These triggers coverage of the activation on the C-John N-terminal kinase, also known as JNK, and nuclear factor kappa B, NFKB signaling pathways. The activation of these signaling pathways promotes the infiltration of monocytes into the AD tissues or AT tissues and their differentiation into the pro-inflammatory M1 macrophages, and by increasing the product of pro-inflammatory cytokines, endothelium adhesion molecules, and chemotatic mediators. The JNK and NF-kappa B signaling pathways, once stimulated, cause adipocytes to produce the necessary pro-inflammatory cytokines, which contributes to insulin resistance and pro-inflammatory macrophage infiltration. The JNK signaling pathway activates pro-inflammatory genes and inhibits the insulin signaling system by reducing the phosphatidylinositol 3 kinase and protein kinase B signaling pathway through insulin receptor substrate 1 inhibitory serine phosphorylation. Activation of the NF-kappa B signaling pathway results in increased expression of numerous NF-kappa B target genes, including tumor necrosis factor, interleukin 6, and monocyte chemotactic protein 1, which leads to serine phosphorylation of IRS1. This prevents insulin signaling. Inflammatory mediators such as free fatty acids, interleukin 6, tumor necrosis factor, and monocyte chemotactic protein 1 circulate in the bloodstream and activate JNK and NF-kappa B signaling pathways in the liver and skeletal muscle. thus blocking systematic insulin signaling.